Here we are again in your subject microbiology, and in this session we are going to discuss the introduction part of bacteria. So expect that we're going to discuss here the different size and compared to other living organism. Also likewise the shapes, arrangement, and the parts of a typical bacterial cell. Under the microscope, these are how your microorganism bacteria could look like. You can see there the different external part of the cell and in reference to the size also of the microorganism. So this is a picture of your prokaryotic cell and eukaryotic cell. This is an animal cell and this is your prokaryotic cell. If you're going to look at, this is more simple as compared to the animal cell. This is a typical animal cell, of course, showing to you the different parts that could possibly found in an animal cell. Whereas in here, this is also an illustration showing to you the possible parts of a prokaryotic cell. But it does not mean that in its prokaryotic cell that you're going to encounter in your study of microbiology have all these parts. Some parts are present, some parts will be absent. And then let us try to differentiate what are prokaryotes and eukaryotes in terms of organism, size, chromosomes, type of nucleus, or what are the nucleic acid present, cell wall, flagella, organelles, and histones. The prokaryotes or the prokaryotic cell versus eukaryotes or the eukaryotic cell. So usually the cyanobacteria or most bacteria and prokaryotes are found under the prokaryotic cell. Bacteria and other higher living forms like the plants, animals, and fungi are your eukaryotic organism. The prokaryotics are smaller or small as compared with eukaryotes. They are even smaller. And then for chromosomes, it has only one chromosome, but for the eukaryotic, they have paired chromosomes. In terms of nuclear or the nuclear characteristics, prokaryotes usually have the circular DNA found in cytoplasm, whereas for eukaryotics, they have the linear DNA found in chromosomes. And then for cell wall, it contains the polysaccharide moraine, but for the case of eukaryotic organism, they do not contain moraine. In terms of flagella, both could probably have flagella, but for the prokaryotic organism, it lacks microtubules as compared with eukaryotic organism. In terms of organelles, it has only very few organelles and some small ribosomes. And then for histones, prokaryotics do not have cistones, but eukaryotic organisms have cistones. So to give more emphasis on their difference, prokaryotic and eukaryotic, both of them are microscopic. However, the eukaryotic are unicellular and multicellular organisms. But for the case of prokaryotic organism, yes, they are microscopic, but they are only unicellular. Prokaryotics lack nuclei and membrane-bound organelles. For the case of eukaryotics, they have both nuclei and membrane-bound organelles. But for the case of virus, they are acellular, not complete cell, Hence, they are also parasitic. They have nucleic acid and a protein. And since they are acellular, they do not have membrane-bound organelles. Or if ever there is, it is limited. So from here, you could see the prokaryotic organism, and this is the eukaryotic organism. And these are illustrations of virus, the AIDS virus and the bacteriophage. So you can see here the main differences. This has more organelles as compared with prokaryotic organisms. And then in terms of dimensions and size, this is your carbon element that could be measured in nanometer. And then here is your tapeworm and the flukeworm, which are measured in meters, or even some molds are also measured in meters. So this is how this looks like, or how big we could see by the naked eye. In terms of yeast, we could no longer see some yeast except for their spores. We can see still some algae in terms of their spores again, or external parts. But in terms of bacteria, they are already measured in micrometer. So your cyanobacteria and like the other protozoan are as small as the red blood cell of human. But there are even some smaller than that. You have the virus. These are illustrations of bacteria under the microscope. So the average size of a bacteria comes in by zero point 0.2 to 1.0 micrometer by 2 to 8 micrometer. So in terms of length, they might be long, but in terms of width, they could be that small. And then this is a table showing to you the difference of size. If you are familiar with this louse, they could be measured in terms of mm. And then for the other microorganisms and component parts of other cells, they come in as big as 100 micrometer. This is for the case of the some algae. And then here is your red blood cell, more or less measured is still in micrometer. And then these are microorganisms already. Some are big as 1 micrometer. But usually most organisms, as you can see here, they are measured in terms of nanometer. And then let's go to morphology, or shapes. The cocci, the bacilli, and the spirillum. Of course, as these are uh, observations under the microscope. So for cocci, they are circular, or they come in by spears. If the microorganism is in rod shape, it is called bacilli. And then if they are in twisted form, they are called the spirillum. There are also some other leumorphic organisms, or they have several shapes. And then the last part is the hyphae. Um, they they are not the same as that of spirillum. The spirillum might only have two twists, whereas for here they have series of twists. So be careful in your observation under the microscope. A hyphae might be interchanged with spirillum, or a series of spirillum, or spirilla. By the way, cocci, the singular is coccus. Bacilli, the singular is bacillus. For spirillum, you have spirilla as the uh, plural. And then here are your examples of rod-shaped organisms. So the first one are illustration of the spears. Here you have the bacilli. So you have here the E. coli with the very long um, appendage or external part. However, these filaments or flagella should not be mistaken as a smaller microorganism. So again, be careful in your observation under the microscope because a part of a microorganism might be mistaken as another microorganism. And then you have the Bacillus anthracis. Then you have here the microorganism responsible for anthrax is Bacillus anthracis. This is how it looks like under the or drawn under the microscope using its observation under the microscope. You have the Fusobacterium nucleatum, and then you have also the Corynebacterium diphtheria for the disease diphtheria. It is still rod shape in a sense that although they have different sizes in both ends, they still form one rod shape. And then you have also the Bacillus, the Corbaceous bacillus, or sometimes they call it the sexy bacillus. So you have here the vibrio, they are comma in shape with some appendages here in this illustration. This is the vibrio cholera. And then we have also the spirillum, possibly in both ends have also flagella, or maybe no flagella at all. You have also here an example of a spirochete. It is more, this is more rigid for us as compared with the spirochete of the disease syphilis, and informing whatever question. Here are the different arrangement of microorganisms. So these are your coccus, the microorganisms which are spherical in shape or round in shape or circular in shape. If they come in as one, they are just simply called coccus or cocci. And then if they come in by two, they are called the Diplococci or Diplococci from the prefix di, meaning two. They could also be in chain or forming a strap. So the microorganism that comes in by two could be in this manner. This is the Neisseria illustration for Neisseria gonorrhea, the microorganism for the disease gonorrhea. But if they come in an end-to-end pairing, they are the Streptococcus pneumoniae, and they are arranged in this manner. So in chain. Okay, some arrangement could be by pockets, not necessarily in a cluster form, but in arranged symmetry. So this is by pocket. So if you're going to divide this, you could see many organisms present. And then if they come in by four, they are called the tetrads. But if you're going to place another plane, you could see that on the upper part is another organism at the lower part. So that is called the sarcinae, or they came in by a cube. As compared with tetrads, they are only by four. So this is how they look like under the microscope. So you could see here just simply 1, 2, 3, 4. 1, 2, 3, 4. Tetrads 1, 2, 3, 4. As compared here, they comes in by cube. So you could see here one cube or one set. That is also one cube. And then the microorganisms could come in by cluster. So they could be in cluster and they are called the Staphylococcus. But if they are in chain, they are called the Streptococcus. Strep. But if they are in stoppable cluster, they are the Staphylococcus. This is Staphylococcus aureus. These are the microorganisms that you have already encountered starting from the previous lecture. So I would like to congratulate you. You have known already some microorganisms in terms of name. So by just simply looking at the names of this microorganism, I am I am quite sure that you know their morphological characteristics, how whether they are coccus, bacillus, or they come in by pairs or they come in by cluster. So let us try to make some exercises. Staphylococcus aureus, what is the shape? Coccus, so it is round. Staphylococcus epidermis, it is round. Is and the Staphylococcus, it means they are in cluster. These are also in cluster. So Streptococcus, from the suffix coccus, they are spherical in shape and they are arranged in chain. This is Vibrio, so they looks like comma. Rhodospirillum rubrum, these are spirillum, so they looks like an spiral. Bacillus subtilis, of course Bacillus, they come in rod shape. Micrococcus luteus, again, they are in spherical formation. So from here to here, maybe you know how they look like already. So congratulations, you count the number of microorganisms here that you can name them already, aside from the other microorganism that we have made mention. Then let's go to the parts of the bacterial cell. The parts of the bacterial cell are quite similar to what you have learned in your biology as parts of the different living organisms. Here is an observation of under the microscope of bacteria, and this is how an artist illustrate for understanding or for learning purposes. So this is this of course refers already to your nuclear area. These are your DNA. The the nuclear area, sorry, the DNA of microorganism might or might not be enclosed. This might not be enclosed with a cover. And then, of course, this is your cytoplasm, and there are many parts here. This includes your ribosome, the plasmid, and then if you go out, you have the plasma membrane, the yellow one, and then we go out again, the cell wall, and then of course the slimy capsule, and then the flagella, the cilia, and the rest. Okay, for the external part, you have the glycocalyx, the flagella, axial filaments, the fimbriae, and the fimbriae. This is a typical flagella. There are different types of flagella actually, and the different flagella allows movement. So this is your bacteria, and those are the flagella. Now, these flagella seems like anchored only to the cell wall, but these are actually made up of different clusters of or group of flagellin, and they are anchored here, and they propel around, causing the movement. So for a more view, here is a bacterial glycocolyx with a slimy nature. So they are usually sticky. They are neatly organized, meaning to say it is not scattering here, but it follows one direction to maintain the shape of the bacteria. And then under the microscope, this is Streptococcus pneumonia, and this part are your capsule there. And then this is again more magnified illustration of the appendage flagella. You have there the flagellin, the hawk, and you have here the basal body. This portion here rotates, allowing the flagella to move in a circular pattern. These are the different types of flagella. At one end, you have the monotrichous. In both ends, you have the amphitrichous. With many flagella at one end is the potrichous. And if the flagella are found in any side, everywhere, you have the peritrichous. Okay, this is also how the flagella move. So uh, the circular motion allows the microorganism to run or to move, and then it opens, and then all will open and stop, and then all this will be clustered again together and allows to propel and then open. Before it opened, it had already moved, so open and then close, and then it will run. There are also some movement caused by axial filaments. The axial filaments produces that uh, circular thing going up movement. So it keeps on turning and turning and turning, but it is actually moving from one place to another. An example to that is the treponema pallidum of the syphilis. The corkscrew motion in the vaginal fluid or vaginal secretion of treponema pallidum make itself transfer from one place to another or one host to another. So here is the illustration. The movement of going there, turning around, going up again, turning around. So that is how the microorganism move. And then here are the fimbriae and the pili. So for the fimbriae and the pili, they are responsible for the microorganism to adhere to the host or to its ecosystem. They they seems like sticky substances, but their ability to adhere itself makes them move from one place to another. Also by the run, move, move, uh, run, open movement. Now the pili and the fimbriae also serves as the site for the transfer of DNA. There are some illustrations that you should know based on the appendages of the cell, and this includes your DNA transfer from one cell to another using the sex pili, that is the reproduction of means of some bacteria. So you have there the cell wall, and then you have the gram positive cell wall. So remember your biochem, these are your peptidoglycans, but in your microbiology, this refers to the set of nutrients responsible in the formation of the cell membrane. This is an example of a gram-positive microorganism under the microscope. Their cell membrane was positively colored with the gram-stained microorganism. That is why it remained the color violet. So here is the illustration. This portion here will allow that the ink or the stain to adhere to its side. Okay, but for gram-negative microorganism, it absorbs the violet or the blue dye, but it maintains the pink color outside the cell membrane. Thus, if you're going to observe that under the microscope, it will look like pink. So here is your gram-negative. So if we say gram-positive, the violet color of the gentian violet is found in the membrane. But if the violet color is negative, the microorganism is gram-negative. So this is the difference between the gram-positive and gram-negative cells. The gram-positive cells contains thick peptidoglycan. For the gram-negative, they have the thin peptidoglycan. Gram-positive microorganisms or bacteria have teichoic acids, whereas for the case of gram-negative cells, they have no teichoic acid. Gram-positive bacteria contains mycolic acid in its outer membrane. As for gram-negative, it is the other way around. So again, you have here the illustration for gram-positive and gram-negative. So you have there the teichoic acid showing to you the pink color, whereas for the at the outside part, whereas for the gram-negative, the dye was absorbed here because of the absence of the wall of teichoic acid. Atypical cell walls have mycoplasma and we have also the archaea. Mycoplasmas lack cell wall. As for the archaea, they are uniquely wall-less. Not only lack cell wall, but wall-less. And then you have also some things to consider in terms of cell wall. Lysosome digests disaccharides and peptidoglycans. Hence, example is the penicillin. It inhibits peptide bridges in peptidoglycan, making it resistant to the symptoms of diseases. This is the plasma membrane. I'm sure you have studied this already. So, here are again your phospholipid layer. This is the main component part of the plasma membrane. You have two phospholipid layer, one here and another here. So this is one phospholipid actually, and these are groups of phospholipid arranged in a manner that the head is up as compared to the other one. Okay, in the laboratory, we will come to apply the gram stain mechanism. So you will be using some crystal violet and iodine crystals in order to come out with the gram positive and gram negative characteristics of both of these bacteria. So the crystal violet iodine crystals do not live in gram positive, but for the case of um the gram negative, the crystal violet iodine staining washes out. So this is the illustration. So for the gram negative, you have here the crystal violet, and then you have there the iodine. Once the alcohol is applied, that will be that determined whether the crystal violet will remain or not. So for the case of gram positive, I have shown you this illustration. The violet or the crystal violet will remain in the phospholipid area, whereas for the case of gram negative, the the pink will just simply predominate. So these are the four steps. You should be guided by the acronym bias, crystal violet, iodine, alcohol, saponin. You will do this. No, I will do this. Because of the COVID quarantine period, you are not allowed to come here. But I hope you will be able to do this when you will come back. Let us try to look for means for you to experience all these things. Okay, I think this is for now. Thank you very much. See you again uh, next time. Okay,